All right, good morning. Today is Wednesday, April 14th. We have hit the halfway point of our fourth section here. So we have finished all of our lectures on the respiratory system, and now we're going to get a week and a half of talking about the urinary system. Obviously a very different system, but we're going to see a lot of similarities because they deal with a lot of similar concepts as well, regulating pH, regulating the movement of materials into and out of the blood, maintaining optimal conditions of the blood, things along those lines as well. Similar amount of assignments for this as we did for the last one. You've got 2 unit reviews. In this case, it's unit reviews 25 and 26. 2 physiology activities. We don't have a home version of this, although I guess you're welcome to urinate in the morning if you'd like, but I'm not going to make you test it or anything like that. But there are 2 physiology, 1 Labster and 1 Physio X exercise that are going to be due. And again, the Physio X, remember, has 6 exercises that you need to deal with for that. And then you got another weekend to study and your Monday lab and lecture exam. And I don't think I'm giving away any trade secrets. Being the smart, sophisticated students that you are fully aware that usually when we have a section like this, there's going to be a daily quiz at some point. So you might want to be aware of when that might be occurring as well. All leading up to our lab and lecture exam, which is on Monday, the 26th. Now, the nice thing about a Monday exam is we don't have that big long 5-day weekend in between moving on from one section to the next section. Usually when we have a Wednesday exam, as you've seen, I've assigned you guys pre-labs and things along those lines to help you to be thinking about the material over that long break before we start the new material. Obviously, we start the new material off after a Monday exam just 2 days later on Wednesday, the 28th. However, one of the big things we're going to be talking about on Wednesday, the 28th, is we are going to be talking about meiosis, and more importantly, really relating meiosis to everything that we learned in 430 on mitosis. And by teaching this class as many times as I have, one of the things that I've noticed is by the end of the 431 semester, which will be pretty much near the end of by that period of time, nobody seems to remember mitosis. So as an encouragement to help you to remember the mitosis things that we've talked about and to prepare for that lecture so that it'll go more smoothly, I am assigning a Labster activity after Monday's exam. So it will be due at the beginning of class on Wednesday, and that will help that Wednesday first lecture go very more smoothly than it normally does. So we'll be able to breeze over some of the basic concepts that hopefully you guys will then remember. Like all Labsters, again, for full credit, you need to do the entire activity and get at least 80% correct. So again, normally there's not an assignment like that for a 2-day break, but this one I found doing this does definitely help with the lecture. So that is our game plan for the next couple weeks. And then after that, we have 4 lectures on the reproductive system, and then exam 5, and then the final exam, and then you are done. So we are rapidly approaching the end. Absolutely, yes, and the crowd goes wild. All right, questions on any of that? Yes, woohoo indeed, woot woot. All right, excellent. So let's stop sharing this and let's bring up our lecture. So we can talk about the urinary system. All righty, so I think I mentioned this once before. Our urinary system is probably one of the worst named of all of the organ systems in our body. But of course, like all organ systems in our body, we have the primary organs and we have the secondary organs. What, of course, are the primary organs of the urinary system?
Kidneys.
Kidneys, absolutely, right? Absolutely not a trick question. These are the rock stars of the urinary system. They are the functional part, right? One of the things that we've learned as we've worked through this is the goal of the body is the goal of life, really, in general. You don't have to just in an A and P, but in any kind of biology class, the goal of life is efficiency. Be efficient, right? It's not to maximize, right? It's about being what is efficient. And life is lazy. As we talked about, once it learns a system, It uses that system as many times as it can and things along those lines, right? But here in our kidneys, we have two kidneys. And if you lose a kidney, does that dramatically change the function of your body? No.
Actually,
Yeah, what if you lost a lung? Would you be able to survive that?
you'd be kind of screwed.
Yeah, but you could live. You probably wouldn't be able to be a professional athlete with one lung, but you could live, but it definitely would impact your life. Heck, even losing one eye. Again, you could live, but you probably couldn't be a professional athlete. Hitting a baseball without that binocular vision might be a little bit more challenging, right? Things along those lines. But could you be a professional athlete with just one kidney? Yeah, in fact, one kidney is capable of doing all of the filtering necessary for the body, right? So you can lose that other kidney, sell it on eBay, whatever it is that you want to do with that other kidney. The remaining one would swell in size a little bit, but it's perfectly capable of doing everything that is needed by the body. There's a tremendous redundancy of this, right? You could be a professional athlete. Now, you probably wouldn't want to be a professional athlete because if anything happened to that second kidney, well, what happens then?
Then you're screwed.
Yeah, exactly. Then you're screwed. Absolutely. Right. Can you survive without a kidney?
No.
No. Right. No, couldn't even be a professional bowler with that. Oh, there you go. Just one. There you go. Yeah, but then you have to worry about your liver if you're a professional bowler because, um, but no, dialysis is a short-term stopgap, but it is not a long-term solution to what it is that the kidneys do. And what is it that the kidneys do? I mean, it's the urinary system, so their primary function must be producing urine, right?
They filter.
Yeah, exactly. Their job isn't about producing urine, isn't like urine is some valuable resource. No, their job is to filter the blood and maintain the conditions of the blood. And it is vitally important that they do that. While you're sitting here calmly in your chair resting, about 25% of your blood is being directed to your kidneys so that they can do this function. It is vitally, vitally important. Yes, in filtering the blood, they produce urine as a byproduct of that, right? And so, in fact, as every single one of us is sitting here, every single one of you is urinating, right? Urinating is the process of making urine, and you're all urinating there as we sit. However, does that necessarily mean that urine is leaking out of your body? Yeah, hopefully not. If it is, then please see a doctor immediately. But, right, we also have our accessory organs known as the urinary tract, which are pretty much responsible solely for the accumulation, storing of that urine until you get to a socially appropriate location, at which point you can then void that urine from your body. And really, that's the key to the urinary tract. It does not modify or change or adjust the urine in any form. It just accumulates it, stores it, and allows for the release in a socially appropriate location. All right. Again, we've talked about this a little bit, but let's be more specific. The job of the kidney is to maintain balance in the blood. Part of that is the removal of wastes and other type of foreign substances. Here we see a large example of biological wastes, many of these involving nitrogen wastes, things like ammonia, urea, which is a metabolic waste from the breakdown of amino acids, bilirubin, which we talked about is from the breakdown of our hemoglobin, creatine, biological waste from muscles, uric acid from the breakdown of nucleic acids, and so on and so forth. But it also helps to maintain blood ionic composition because remember, whatever happens in the plasma of the blood affects the interstitial fluid. So by maintaining the condition of the blood, we maintain the conditions of the body. So we need to make sure that we have high levels of sodium, low levels of potassium, appropriate amounts of calcium, and so on and so forth. Maintaining those ionic balances so that our muscles can function properly, our neurons can function properly, and so on and so forth. Right? We have to maintain an appropriate blood volume. Right? So the amount of water in the body is vitally, vitally important that we have. And as we know, the volume of the blood in our blood vessels also affects what?
Heart rate, blood pressure.
Blood pressure is the one you're right. I was looking for, but you're right. Heart rate as well. But absolutely, blood volume basically affects blood pressure. The more water that's in the blood vessels, the higher the blood pressure is. In fact, again, if you have high blood pressure, one of the first things they do is put you on a diuretic. What does that diuretic make you do?
Make you want to pee.
True, it makes you pee a lot. Yeah, that's true. Absolutely. It flushes water out of your body. And so blood volume drops, blood pressure drops as well. So there we have this direct effect on blood volume, which has an indirect effect on blood pressure. But a kidney also produces hormones and hormone-like proteins like renin that also directly affect blood pressure as well. So our kidneys both directly and indirectly influence blood pressure. That isn't enough. Our kidneys play an important role in maintaining blood osmolarity. What do I mean by osmolarity?
The amount of solutes to solvent.
Absolutely. It's basically the amount of stuff that is in the plasma of the blood and therefore in our interstitial fluid. And we do mean stuff, right? If I were to take 1 mole of sugar and put it in 1 liter of water, and I were to take 1 mole of salt and put it into 1 liter of water, would those have the same osmolarity? What do you
No.
think?
Yes.
Right. Well, so okay, so I've heard both yes and both no. However, let's think of it this way, being the smart, sophisticated students that you are. If the answer was yes, would I be asking this question?
I suppose it depends on your mood.
True. Although
Very tricky one.
usually I try to be clever. So in this case, I'm trying to be clever. And so the answer, of course, is no, they're not the same. So someone explain to me why.
Because the molecules of the sugar are bigger and exert more osmotic pressure on water than salt does.
That is a great guess. However, when it comes to osmolarity, the size of the stuff doesn't actually matter. Right? It's like when you pick up your mail. Your mailbox has 10 pieces of mail in it. All of those mail aren't necessarily the same size, but they all still count as 10 things. So that tiny little postcard that you get from grandma and that big, huge, mailer telling you that, you can get a great rate on home insurance or something like that. All of those count the same. So why are these not the same? Size isn't what matters in the case of osmolarity. It really is just a count of the amount of stuff. So why are these two things different?
Sugar associates into more things,
Does sugar break up into more things?
or salt breaks into more things.
Okay, so when I add sugar into water, that glucose, for instance, right, as we know, just stays a single molecule. And so let's go with an easy one. I add 100 glucose to this. How many things do I have inside of this beaker?
100 things.
100 things. When I add 100 sodium chloride to this thing, to this beaker, what do I get there? How many things do I have now?
100 Na and 100 Cl.
Ah, exactly, because salt disassociates. So what happens is that salt separates into 100 sodium ions and 100 chloride ions, giving us a total of 200 things. Osmolarity doesn't care whether you're sugar, whether you're salt, whether you're sodium, whether you're a nucleic acid. It's just a count of the amount of stuff. And so, yes, that salt water has twice as many things than the sugar water, and it has twice the osmolarity. So osmolarity really is just a count of the amount of stuff, how many things are present. And as we here in Sacramento know better than most, although again, as the years continue to go on, this example seems to be less and less relevant. But if you were to drink massive amounts of water in a very short period of time to win what is now an incredibly antiquated video gaming system, you dramatically upset the osmolarity by adding all that water, changing the balance of amount of things that are in the solutions of your body, which in your blood plasma, which affects your interstitial fluid, leading to cell death, leading to organ failure, leading ultimately to death, right? Just as long as you don't wee for the wee. Right. So obviously that osmolarity is incredibly, incredibly important. All right, excellent. And much like we saw in our respiratory system, we know that we can influence blood pH. Now, what would it be? Obviously, carbon dioxide and regulating carbon dioxide levels is how our lungs control the pH. How would our kidneys control the pH?
Secretion of bicarbonate ions.
Exactly. Not just bicarbonate ions, but hydrogen ions as well. By hydrogen ions and bicarbonate ions. Right? If we can regulate the amount of hydrogen and bicarbonate, we can influence our blood. And I think I actually say that right there. There you go. Excellent. So, right, hold on to more hydrogen ions will make the blood more acidic. Get rid of more hydrogen ions, make the blood more basic. Hold on to more bicarbonate, and we have more of a buffer that helps to stabilize and maintain the pH. So by regulating the amount of hydrogen ions and bicarbonate, we can directly influence the pH of our blood. Like I mentioned, our kidney produces a handful of hormones or hormone-like proteins. Some of the hormones that are produced or activated in our hormones were erythropoietin. Someone remind me again what erythropoietin does?
Stimulates blood cell growth.
And not just any blood cell, but which blood cell?
Red blood cell.
Red blood cells, absolutely. Remember, erythropoietin makes those red blood cells mature more rapidly, and you produce more of them. Calcitriol is activated in the hormone. Calcitriol is the active form of what again?
Vitamin D.
Vitamin D. Excellent. And what did calcitriol do?
It helps absorb calcium.
Yeah, it tells the digestive system to absorb calcium. Excellent. Absolutely. And there are others as well. And then lastly, in extreme cases, and this can occur, for instance, again, happy Ramadan for those of you who are celebrating it. But in cases of fasting, one of the things that our kidney is actually able to do is actually break down our amino acids, primarily glutamine, to actually make glucose. So it can actually help to maintain blood glucose levels in cases of fasting and things along those lines. In a process known as gluconeogenesis, right? Neogenesis to give birth to bring to form, to bring to light glucose. So making glucose primarily by breaking down the amino acid glutamine. So this is a common process that occurs, like I said, during times of fasting, like right now during Ramadan. So very appropriate for what's going on right now. All right, excellent. So, like I said, I wasn't kidding. The kidney is truly a rock star, producing and providing a tremendous number of important functions for us. All right, questions on that? All right, excellent. So let's talk about the kidneys. They are located in the posterior portion of the abdominal pelvic cavity, and our kidneys are part of the sad puckers that are retroperitoneal. Someone remind me again what that means?
not in the peritoneal.
True. Okay, absolutely. But so where is it in relation to the peritoneal cavity?
behind it.
Yeah, behind it, absolutely, right? The parietal peritoneum that lines the wall of the abdominal pelvic cavity drapes over the top of, as you can see here, most of the structures we see here. The kidneys, the aorta, the inferior vena cava, the adrenal glands, the ureters. Remember, all those things we talked about are behind the abdominal pelvic cavity, behind the parietal peritoneum. So we see that nicely there. Here in our cross section, we get a better appreciation of this. So again, here we see that parietal peritoneum lining the wall of the abdominal pelvic cavity. Here is where our intraperitoneal organs, like the stomach, like most of the small intestines and so on and so forth are located. And then here behind it, we see anchored in its little pockets behind that, we see our kidneys to the two sides. Now notice, as I mentioned, it is indeed inside of a pocket. Just because it is not inside the abdominal pelvic cavity doesn't mean it isn't protected. So let's actually grab my highlighter. What color will show up well? Maybe the purple will show up well. Notice one of the things, obviously the outer surface, and let's actually make this smaller. The outer surface, as we talked about, is that parietal peritoneum lining the outer surface of it, covering the outer surface of it. But notice underneath that is this fibrous pocket. This fibrous pocket is what is known as the renal fascia. And notice it truly does form a pocket by having both an anterior fascia and a posterior fascia. So we have these two layers, the anterior renal fascia and the posterior renal fascia, that form these two pockets that contain the kidneys inside of them. However, notice the kidneys aren't just sitting in there rattling around inside of those pockets. Those kidneys have this nice, thick, cushioned padding, right? Kind of like those shipping peanuts wrapped around it, providing that protection. Of course, we don't have anatomical shipping peanuts, but what we do have is a thick layer of adipose, what is known as our perirenal, because it is around the kidneys, fat capsule. This helps to cushion and support and protect the kidney in space. Sponsored by Cheetos? Oh, at the fat capsule? This fat capsule is vitally important. Again, as we've talked about in the past, we always think of fat as being bad, right? Right around the corner, we've got summer, so we all have to get into our bikini body attire. Body shape. So, of course, like me, I'm sure you were all on the popcorn diet, right? I'm on the popcorn diet right now, where I have one piece of popcorn for breakfast, one piece of popcorn for lunch, and a very reasonable 3 pieces of popcorn for dinner in the evening, right? Now, if I had a diet like that, am I going to lose weight as a result of that? Yeah, because I'm going to be starving myself. Now, as we've talked about before, I'll start losing muscle first before I start losing fat, because when you starve yourself, your body holds on to fat. But when you look at these people who go on this extreme weight loss, the Biggest Loser TV show, things along those lines, or some of the surgical ones where they do like the stomach banding and things along those lines, where they lose, like what's his face, the weather guy on the Today Show went on.
Al Roker.
Yeah, there you go. Exactly. When people have those types of extreme weight loss, rapid extreme weight loss, one of the things that their doctors monitor very, very closely is their urine output. Because while we see all the fat melting away on the outside, the problem is you're losing fat on the inside of the body as well. And fat is a very dynamic tissue, constantly producing triglycerides, bundling them up and moving them around. So with moderate weight loss, you're able to redistribute fat to where it's needed and away from the areas where it's more excessive. But when you have that extreme weight loss, you lose all the fat all at once. And so one of the things that can happen, if you think about it, we have this kidney that is sitting inside of your abdominal pelvic cavity, connected by the ureter to the bladder underneath. And what happens is that if you start to lose a fat very, very rapidly, that adipose capsule around the kidney starts to go away. And what actually can happen is that the kidney can start to descend in the abdominal pelvic cavity. It is a condition we call ptosis of the liver. I mean, of the kidney. And the problem of that ptosis of the kidney, the problem that occurs when that occurs, if you think about this, is it descends, it can cause a kinking in your ureter. And when you're watering your garden out in the back and you kink the hose, what happens?
Flow, slows, or stops.
Flow slows or stops, and that water and the pressure of that water backs up into the kidney and can cause severe and in some cases permanent damage to the kidney. So like I said, when there's these people that are surgically doing it like Al Roker, or those people who are on like the shows like The Biggest Loser and things along those lines, their doctors are monitoring their urine output very, very closely to make sure that they're not dealing with this issue of ptosis, because like I said, it can be extreme and permanent damage that can occur to the kidneys because of that.
So how do you fix that kinked ureter or that descending kidney?
If it occurs, then they actually what they will do one of two things. They will either go in and surgically support the kidney in place, elevate the kidney in place, or they can go into the ureter and put a stent in it so that it doesn't kink in that particular location. So typically it takes those types of extreme types of resolutions to do that. So that's one of the reasons why, again, it's monitored so closely so that that damage doesn't occur. All right. We know it's retroperitoneal, and I think I need to move my pictures a little bit so we can see this. But one of the other interesting things about the kidneys is they are asymmetrical in their location. While both kidneys are partially protected by the ribs, because of the ribs, the way they descend down. If you notice, the right kidney is slightly inferior to the left kidney. And why might that be the case?
Because of the liver?
Yeah, that largest visceral organ, the liver, basically shoves the right kidney down a little bit more inferiorly in the abdominal pelvic cavity. So we have a slight asymmetry. And again, especially from body blows or posterior blows, as you can see, the right kidney is slightly more likely to be injured because of that slightly inferior location in the abdominal pelvic cavity. Now, kidneys have that very stereotypical kidney shape, as we've seen before. We saw it in the lungs, we saw it in the lymph nodes, we saw it in a lot of different places where it has a convex surface and a concave surface. And as we've talked about before, that concave region, the indented region, we identify again with that term, the hilum. And much like we saw in the lungs, the hilum is the location where the blood vessels, the nerves, and the ureters are all going to feed into and out of the kidney. So everything feeding into and out of the kidney enters through that hilum, similar to what we saw in the lungs. Let's take a look at the anatomy of the kidney, starting first with the gross anatomy. Here we see the structure of it with that convex and concave surface and the structures entering into it. We have three primary regions to the kidney. And I guess actually we should go back one more step as well. When we're talking about the protections of the kidney, we have that anterior and posterior renal fascia. We have the fat capsule that wraps around it. And notice also we have a thick fibrous outer coating to the kidney as well, what is known as the renal capsule or fibrous capsule. So we have that fibrous capsule there. That is that outer portion. Then, as we've seen in many organs, like the lymph nodes, like bone, like lots of other places, we have an outer region and an inner region. What do we call the outer region?
Cortex.
Cortex. So notice we have this cortex that wraps around here. This illustration doesn't do as nice of a job of showing it, but we'll see it in the actual kidney in a minute. This cortex is a very granular tissue with a massive amount of blood vessels located in it. And then while we have a cortex, we also have the inner region, which is going to be referred to as what?
Medulla.
The medulla. The medulla is the inside region. However, one of the distinct characteristics of the kidney is that we don't just have a singular medullary region. What we actually have is somewhere close to a dozen individual pyramid shape central regions. So these are structures are known as the medullary pyramids. Notice one of the things that distinguishes the medullary pyramids is that they're actually extensions of the cortex that separate the kidney into individual compartments. These extensions of the cortex are what are known as the renal columns. And they actually divide the kidney into individual lobes. So a lobe is basically the pyramid and the cortex that surrounds it. So that last one didn't look quite so good. Let's do that. So this compartment right here, the medulla, and that cortex that surrounds that medulla is what we distinguish as an individual lobe. So again, oh, and that's the other thing. The cortex of our kidneys contain the functional units of the kidney, and that functional unit of the kidney is what is known as a nephron. It is that nephron is what is responsible for filtering the blood and doing all of those functions we just finished talking about for the kidney. Those 8 functions all happen there in our nephrons. As I mentioned, we have our medulla. Get rid of that. Again, I mentioned about a dozen. It can vary from person to person, 6 to 18 of these individual pyramids. Again, while morphologically, some people have as few as 6, some people have as many as 18, there doesn't appear to be any difference in the function of the kidney. So it's not like more pyramids mean the kidney is more efficient or anything like that. It really just seems to be some morphological variance, but it doesn't affect the function as far as we know. One of the interesting things you'll see, and again, they don't do as nice of a job of distinguishing it on this particular illustration, but we'll see it in the real kidney in a moment, is that there are these, that doesn't show up as all at all. There are these deep grooves or striations in the pyramids going from the base to the apex of the kidney. And notice the apex of the kidney actually sticks out in a little finger-like extension known as the papillae of the pyramid. So there's a little papilla at the end that sticks out at the apex of our pyramids that all these stripes basically direct for, and that's because this contains massive amounts of tubules and blood vessels that are all oriented in the exact same orientation, giving the pyramids a very striped appearance. And we just talked about how they're divided into the columns. And if you think about it, if someone has 6 to 18 pyramids, then as we mentioned, they're going to have 6 to 18 lobes. 1 lobe for each pyramid with the cortex that surrounds it. All right. Now, notice there is one additional region. I'll use purple for this one. This third region is actually a space. So notice where the hilum enters, the kidney actually has a hollow space. And this hollow space is known as the renal sinus. Right? Sinus is a space. That's a term we've used before in this class. Now, notice this space isn't completely empty. There is a lot of stuff in this space. Let's start with some of the easier stuff. Notice that our renal capsule, that fibrous capsule, does extend into and forms a boundary inside of this space. So that renal capsule, that fibrous capsule, is going to be found here lining our renal sinus. Notice some of our adipose from the fat capsule extends into this space as well. So we can see that adipose entering into our renal sinus. And then obviously our large blood vessels and nerves are going to be located in here. But the other thing that we have here in our renal sinuses are the collecting structures. As we talked about up here, I'll use black for this. We have those nephrons, we have these tubules. All of this is where we are processing the blood, filtering the blood, pulling the stuff out, making the urine. Once that fluid is released from the apex of our pyramid, from that papillae, it is now officially urine. And at this point, it is not processed anymore by the body. So instead, we just need to collect it and carry it to the bladder to be stored until we're ready to release it. So basically what we have are these collecting vessels. These collecting vessels basically form three main structures. The first is what we call a minor calyx. Notice the key to a definition of a minor calyx is that it receives urine from just one papillae. So notice, and I'll color it here with the highlighter, this tubule essentially right there basically just collects urine from this pyramid. Notice this one here collects urine from just this pyramid. So both of those would be examples of minor calyces. However, if you look right here, this piece of tubule gets urine from both this and this minor calyx. So it's getting it from at least two pyramids. So that would be considered a major calyx. So major calyces, that's the plural, major calyces receive urine from more than one pyramid, whereas a minor calyx receives urine from just one papilla. Lastly, these major and minor calyces feed into a singular funnel-shaped structure, and this singular funnel-shaped structure is known as the renal pelvis. And the renal pelvis is what carries the urine out of the kidney and feeds it into our ureter, whose job it is to carry that urine down to the bladder. So if we were to follow the path of our urine, it is expressed out of the papillae of a pyramid into minor calyx, into major calyces, into the renal pelvis, and finally into the ureter. Questions on that? All right, we have taken advantage of this pretty illustration from your textbook, but notice we can see this nicely in an actual kidney as well. First, we notice the cortex and those renal columns. Notice we have the pyramids here. And again, notice as you look at these pyramids, you can see the striations, the stripes in them from all the tubules that are going in those. Let's take a look at this one here, because this one's a nice straight example. Here is a single pyramid feeding into a single minor calyx. Receives just from there. Notice it is then feeding into a major calyx here. So big M for major, because notice it's receiving from this minor and from that minor. It feeds into the funnel-shaped pelvis, which collects all the urine from all of the calyces, brings it out of the kidney, and feeds it into the ureter. All right, if we were in the classroom today, we would be actually dissecting pig kidneys, and you'd get a chance to actually see all of this. All right, questions on that? All right, now notice what we're not seeing here nicely, although we see a little bit of it, or we didn't see at all in the illustration, is we see a little bit of the blood vessels and none of the nerves. But the kidney has both a very elaborate neural and blood vessel structure to it. Now, again, while you're sitting here at rest, 25% of your blood is being filtered by your kidneys. This is a vitally important process. All right, so the nerve supply via the renal plexus. And it has an elaborate autonomic nerve, fiber, and ganglion structure associated with it, its own little renal plexus. Does our does our kidney receive dual innervation? Does it get equal input from both the sympathetic and the parasympathetic? Well, again, I'm asking the question, so what's the obvious answer?
No.
No, because if it did, then I wouldn't be making a point of emphasizing it. Exactly, no. In fact, the kidney, if you remember correctly, way back in 430, we talked about how most of the organs of the body have dual innervation, get equal input from both the sympathetic and parasympathetic, but there were some examples of things that don't. And for our kidney, it primarily receives input from what branch of the autonomic nervous system? Remember, we have the two branches. I'm sorry, say again?
The parasympathetic?
Parasympathetic, absolutely. Because if you remember, if you think of in terms of parasympathetic, that is our rest and digest. Our sympathetic is our fight and flight. So when we think of filtering the blood, that definitely sounds more like a housekeeping type of activity than a fight or flight type of activity where we need to use energy. So of course, it is going to be primarily innervated by the sympathetic nervous system. All right, questions on that? Wait, what? You don't have a problem with that? All right, Lori's got a problem with it. Absolutely, right? Sympathetics are fight or flight. It doesn't seem like it would make sense that this should be innervated primarily by the sympathetic nervous system. That seems like a parasympathetic type of activity. It turns out, though, it's a little bit more sophisticated than that. We will actually talk about the neural control of the kidneys, but let's talk about it this way instead. While you're at rest, as I talked about, 25% of the blood is going to the kidneys. As a result of that, we have a massive blood volume going to the kidneys. And that massive blood volume is going to give us a massive blood pressure. And as we know, pressure is what makes the world go round. Now, filtering the blood is something we could only survive 1 or 2 days without having it occur. So do you think it's absolutely necessary for our parasympathetic nervous system to say, come on, kidneys, do your job? It's important for you to do your job? No, probably not. It probably doesn't need that type of encouragement. However, when we're exercising, when our sympathetic nervous system, or when, let's say it's active, it doesn't have to be exercising, when we're active, right? We're on a 15-minute break and you have to run downstairs and make your coffee and make a quick sandwich and eat that and run back upstairs. When you're active and you're moving around, does more blood or less blood go to your kidneys? When you're jogging on the treadmill, do you still have 25% of your blood going to your kidneys?
Less
No.
blood.
Yeah. So when active, less blood goes to the kidneys, right? If we have less volume, we have less pressure. And if we have less pressure, is the kidney going to be able to do its work as efficiently as it was when you're at rest? No. So every time you get up and walk around, do you want your kidneys to function less efficiently than they were when you were seated? No. So even though there's less volume of blood, even though there's less pressure, we still need the same level of efficiency. So when active, the sympathetic nervous system gives the kidneys, and for here, we'll just say a boost. We will talk about this control and how it's important and how it works in more detail on Monday. But here we can at least start to understand why. So again, it may not seem intuitive, but as we'll see, this sympathetic control when we're active is going to help us to compensate for the fact that less blood is going to the kidney. So less blood means less pressure, means less efficiency, but we want to maintain that efficiency. And so our sympathetic nervous system helps us to do that. All right, and it comes off our celiac plexus, as does, remember, our phrenic nerve goes to that, and again, controlling our diaphragm. We talked about that. All right, and like I said, the whole point of this is to maintain appropriate blood flow because the volume affects the pressure. And pressure, as we know, is what makes the world go round. So we can influence urine formation and maintain appropriate filtering of the blood even when active. That's the goal. We want to maintain the efficient filtering of the blood even when active. Excellent. All right. Now, as I mentioned, we have an elaborate neural structure associated with our kidney. We also have a very elaborate blood supply to the kidneys. After all, if 25% of the blood is going to be going to the kidneys while we're at rest, we need an elaborate blood vessel structure to accommodate that. And like I said, the kidneys are indeed a workhorse. Out of that blood, our kidneys filter 200 liters of fluid that we call filtrate from the blood in a 24-hour period of time. Notice I didn't use the word urine because do we produce 200 liters of urine during the course of a day? No. For starters, you don't have 200 liters of extra fluid you could afford to lose during the course of the day. But more importantly, if you produce 200 liters of urine during the course of the day, we'd all be sitting in the bathroom right now during this lecture, right? And that's obviously not the case. So while we produce 200 liters of filtrate, 200 liters of stuff, of water and stuff, are filtered out of the blood every day. Over 99% of it is reabsorbed back into the blood. So while we produce over 200 liters of filtrate, during a typical day, we only produce about 1 to 2 liters of urine. So there's a massive movement of stuff out and then a massive movement of stuff back in. Very, very busy, busy process. So again, not surprisingly, we have an incredibly elaborate blood vessel structure associated with this. Let's actually talk about this. Let's draw it. How are we on time? We can do this quickly. Excellent. All right. So first thing we need is a kidney. Excellent. And there is our sinus of the kidney. Here I'll put some pyramids with their papillae sticking out. That works. Excellent. So let's start first with our arteries. Oops, didn't spell that right. Oops, still didn't spell that right. There we go. Give me the path of blood from the heart to the kidneys.
Left atria, left ventricle, into the ascending aorta, the aortic arch, descending aorta, thoracic aorta, abdominal aorta, and it splits off to the renal artery.
Perfect. Let's start there. So again, absolutely all of that is correct. Although I would say that you don't have to say both descending and thoracic and abdominal, because remember the thoracic and abdominal together make the descending, but you've got absolutely the right idea. So let's start first with the abdominal aorta. And then from there, as you mentioned, we enter into the renal artery. The renal artery is what actually travels to the kidney, but doesn't actually enter the kidney. So actually, let's do it this way. So this is the renal artery, and I'll just put A there for simplicity. As the renal artery comes towards the kidney, it then branches. And it is actually the branches that enter into the hilum. And these branches are what are known as the segmental arteries. So the segmental arteries are actually the branches that enter into the hilum of the kidney. From there, the segmental arteries branch, and as they branch, they form these blood vessels that go between the lobes of our, here, let's draw one more here. These go between the lobes of our kidney. And guess what we call the arteries that go between the lobes of the kidney?
Interlobular.
Interlobal.
Yeah. Interlobar arteries. Excellent. So we have our interlobar arteries. Those interlobar arteries then branch to arc, over the base of the pyramids, basically helping to form the boundary between our medulla and, I want that to be black. Between the medulla and the cortex. So we have these arching, or one might even say arc-shaped, curved blood vessels that arc over the top of the base of the medulla. How do you think we identify those? If only I had a list of the anatomy I was responsible for on this class. Make it so much easier to answer his questions.
You make that wish a lot.
I do make that wish a lot. And one day it might
Arcuate.
actually come true. Say again?
Arcuate.
Yeah, exactly. The arcuate artery. Excellent. Excellent, excellent, excellent. Those arcuate arteries then feed arteries that extend out, one could even say radiate out into the cortex. And how do you think we identify these blood vessels that radiate out into the cortex?
Cortical radiate artery.
There you go. Now, these cortical radiate arteries are also sometimes, because they are contained within the lobes, are sometimes referred to as the interlobular artery. You may see that term as well, but I think our anatomists, as much as they hate you and as mean as they like to be, I think realize that interlobar and interlobular were about as confusing as it possibly could be. So more commonly now you will see them referred to as the cortical radiate artery, but interlobular is certainly acceptable because it is an appropriate term for those. So notice if we come back over here to our list, our renal artery feeds into our segmental. Oops, I need to make this big again. Artery, which feeds into our interlobar artery, which feeds into our arcuate artery, which feeds into our cortical radiant artery. Excellent. So we have these arteries leading to, and that cortical radiant artery is then going to feed into our nephron. Remember, our nephron, we said, was that functional structure. As we'll learn, the nephron contains not one, but two capillary beds. Let's just say capillaries in it. And of course, as we know, feeding into capillaries are going to be arterioles. So those cortical radiant arteries feed into arterioles. Yeah. There we go, into our arterioles. So let's draw a tiny little arteriole here, and then we will draw our box in purple. That is our nephron. We'll talk about the blood vessels of our nephron in just a second. Oh, and I changed the color of that. So let's go back and cheat. We'll talk about what's happening in our nephron in just a second. But before we do that, let's talk about the veins coming back. So again, this is the path of the blood in our arteries coming down to the nephron. And then from our nephron, we have the veins coming back. And luckily for us, there is a fair amount of symmetry. Starting first, feeding out of our nephron is going to be a venule. And that venule is going to feed into veins that radiate down from the cortex towards the border between the medulla and the cortex. And guess how we would identify these small, one could even argue, radiating veins coming out of the cortex? What do you think? Well, if they were cortical radiant arteries, guess what the veins radiating out of the cortex are called? I did mention there was symmetry, right?
Cortical radiant vein.
There you go. Those cortical radiant veins feed into veins that basically help to define and run along the border between the medulla and the cortex. What do you think we call these arcing veins that radiate over? I mean that arc over the top of the medulla.
Arcuate veins.
There you go. Yeah, this should be getting easier as I'm going through this. Those arcuate veins feed into veins that travel in the space between the lobes of our kidney. What do you think we call these veins that travel down between the lobes of our kidney?
Interlobar veins.
Again, I thought this might be a little bit easier because if you've noticed, there's some symmetry here. We have, and I'll cheat and do it this way. So notice we have this complete symmetry as we're working our way up. We went, right, renal artery into segmental artery, into interlobar artery, into arcuate artery, cortical radiant artery, arterioles, nephron, venules out, cortical radiant vein, arcuate vein, interlobar vein. So of course, what is next?
Segmental vein.
Exactly, except for one problem. What do we know about anatomists?
They hate us.
Hate us.
They hate you. And so there are no segmental veins. Instead, all the interlobar veins come together. And as all the interlobar veins come together, they come together to form the renal vein. So it turns out there is no segmental veins. Instead, interlobar veins just feed into the renal vein. Which, of course, as you know, feeds into what? What does the renal vein feed into?
Inferior vena cava.
There you go. There you go. So no segmental vein, but other than that, we have symmetry. All righty. With me so far? All right, excellent. We will talk about the nephron in much more depth in a moment, but I want to connect our blood flow here. So as I mentioned, there are 2 capillaries in our nephron, and in fact, there are 3.5. Blood vessels that make up a nephron. So the first of these is a very, very specialized capillary known as a glomerulus. Let's do it this way. One of the ways that the glomerulus is special is it is fed into and out of by an arteriole, right? If we think of most capillaries, an arteriole feeds in, a venule feeds out. But that is not the case here. Here, the glomerulus has an arteriole that feeds into it and an arteriole that feeds out of it. So one of the things we need to do is to be able to tell these apart. One of the ways we can tell them apart is by giving them names. One arteriole feeds into the glomerulus and one arteriole feeds out. So if I think back to some of my anatomical terminology, like for instance, a lymph node or like my nervous system, when something feeds into it, how do we refer to it usually?
Afferent.
Afferent. So our afferent arteriole feeds into the glomerulus. And notice that afferent arteriole is not part of the nephron. However, the one that feeds out of the glomerulus, which of course, as Allison pointed out, is the efferent arteriole, is considered part of the nephron. So blood vessel 1. Blood vessel 2. And that efferent arteriole feeds into the second capillary. This second capillary is called the peritubular capillary. It is a much more traditional capillary. And it is this peritubular capillary that feeds into the venules that feed out of the nephron. Now notice I did say 3.5. So what we'll do here is throw in a 3A. Sometimes there is a specialized additional capillary here. And that special capillary is called a vasa recta. Again, we will talk about these in much, much more detail, but some nephrons have them, but not all. So we'll put it as a 3A. We'll put it as a half because some have a vasa recta, some don't. And we'll talk about that distinction in a bit. But I wanted to be able to put this all together so we could see the entire flow of blood. Afferent arteriole feeds into the glomerulus. Actually, let's do it this way. Glomerulus feeds into the efferent arteriole, feeds into the peritubular capillary or the vasa recta. And then either the vasa recta or the peritubular capillary feeds into the venule. So we see the complete blood supply. And again, I've done a truly amazing job of drawing this, but let's notice that your book's got some pretty pictures that show this as well. Notice here we see the arteries flowing in from the abdominal aorta. We feed into the renal artery, which feeds into the segmental arteries, that feeds into the interlobar arteries, that feeds into the arcuate arteries, that feeds into the cortical radiant arteries, that feed into, even though it doesn't show it here, oops, that afferent arteriole. That afferent arteriole feeds into the nephron. And like I said, we'll talk about these blood vessels in much more detail later. They include the glomerulus, the efferent arteriole, and the peritubular capillaries, which sometimes include the vasa recta. And then it feeds out into the veins, venules, into cortical radiant veins, into arcuate veins, into interlobar veins. But notice the interlobar veins, for whatever reason, they don't call these segmental veins. Why these are segmental arteries, but this isn't a segmental vein, I have no answer for you, other than the fact that, as we pointed out before, that anatomists hate you. But these interlobar veins feed into the renal vein, which of course feeds into the inferior vena cava. So there you go. There is the entire blood vessel structure of our kidney. And I guarantee on a model and a chart on an illustration like this, I will have pictures of this pointing at each of these individual blood vessels, and you absolutely positively have to know them. As well as I could always ask the essay question of you describing the blood flow through the kidney, and you would have to know these in order as well. So know your driving directions, know your structures. All right. Questions on that? All right, excellent. That brings us to that nephron. Nephron's going to take a lot of work. It is the functional unit of the kidneys. Each kidney has over 1 million of them. And the kidneys are a combination of both tubules and blood vessels. So we have named the blood vessels, but we haven't talked a lot of detail about them yet, but we will do that. So we'll go over the names of the blood vessels again. We will identify the tubules. We'll put all the pieces together. And after the break, we will build a nephron. All right, so that will be our game plan. We will build a nephron, but we will do that after the break. So let's go ahead and take our first break. Looks like it's 9:16 now, so we will return at 9:31. And at 9:31, we will pick up from there. And I will start the recording. All right, any questions before, any questions before we take our first break? All right, see you in 15 minutes. All righty, hopefully you're caffeinated. We're getting there. And let's go ahead and get started. So our goal is, like I said, to talk about the nephrons. Each kidney, and again, I'm not the one who did the counting on this, but I'll take their words for it, has over 1 million nephrons made up of blood vessels that we talked about, and also these tubules for processing our blood, for processing that filtrate that we talked about. And if you were to take all the nephrons and lay them out end to end, it would hurt a lot. Right. As we mentioned, this is responsible for forming our urine. But again, it's the urinary system. We're forming the urine, but we've got that asterisk. Obviously, the urine isn't the important part of this process. The process is about filtering the blood. Yes, when we filter the blood, the things we don't want or don't need come out in the urine. And that's useful also from a clinical standpoint, because if there's something wrong with you, one of the first things you do is pee in a cup, because what comes out of you tells the doctor something about what's going on inside of you. But like I said, the real goal is to maintain appropriate homeostasis of the blood. And like I said, nephron is made up of two parts, blood vessels and renal tubules. Now let's first look at this pretty picture right here. And we can talk and then switch to the whiteboard to talk about the anatomy. Save this, clear that. And we can talk about our nephron. Now, as I mentioned, the nephron is comprised of renal tubules and blood vessels. We have mentioned the names of our blood vessels, so let's go ahead and write those down. Again, we talked about the glomerulus. And we know that glomerulus, as I mentioned, is a specialized capillary. Yes, no, this is not you've got mail. You've got urine. That's what this is basically, yeah. Luckily, I don't know any other norepinephrine. Oh, wait, Sleepless in Seattle. There you go. I don't know any others than that. All right. Glomerulus, a specialized capillary that, let's give ourselves some more room. Let's do it this way. Excellent. Second is the efferent arteriole. Third is the paratubular capillaries. Let's just be singular. And the fourth is, or 3A, vasa recta. And we'll go ahead and call it 4. All right, so those are our 4 blood vessels. And conveniently enough, there's 4 renal tubules as well. The first renal tubular structure is what is known as the glomerular capsule. Because guess what it wraps around? Not your question.
Around the glomerulus?
Around the glomerulus, absolutely. Of course, the glomerular capsule was first discovered by good old Bob. And what was Bob's last name? Bowman? No one's seen it. Also sometimes referred to as Bowman's capsule. Right. Now, this glomerular capsule or Bowman's capsule is actually a double layered structure. And actually, let's do this because I want more room. So we can switch down to this. Oh, no, I want that big. And then so not surprisingly, with a double layered structure, we have a parietal layer and a visceral layer. So let's do a little drawing. We'll talk about the anatomy. Oh, actually, let's start with this. This, of course, is our afferent arteriole. Which again is not a part of the nephron. That's why I colored it pink to remind us that it is an artery feeding in, but it is not a part of the nephron. And what it feeds into is our first blood vessel, which is again that glomerulus. And the glomerulus is this big, huge coiled capillary. So what happens here, and I guess we'll use green, is Bowman's capsule, or more specifically, the parietal layer of Bowman's capsule, is a simple squamous epithelial tissue that basically just forms a cup whose job it is to catch that filtrate as it comes out of the glomerulus. Again, remember, as we talked about, we produce 200 liters of filtrate during the course of the day, and we need some cup to be able to catch that with. And that's basically what the parietal layer of Bowman's capsule does. It captures this fluid as it is being released from the glomerulus. Now, as I also mentioned, there is a parietal layer, a visceral layer. The visceral layer is directly in contact with the glomerulus, lining the surface of the glomerulus, helping to support this specialized capillary. And it is made up of specialized cells as well. A parietal layer, as I mentioned, is simple squamous. Its job is basically just to be a structure to cup the catch. And so by being a simple squamous, it's small, which is important because we need to fit 1 million of these into a single kidney. The visceral layer helps to filter the filtrate that comes out, and it is made up of these special cells called podocytes that are these specialized cells that are going to play an important role in helping to regulate and filter the plasma that comes out. They're called podocytes because they have these long foot-like processes that come off of them that interdigitate with each other and form a filtration slits that, again, limit the movement of stuff out. Now notice Bowman's capsule and the glomerulus do not touch. There is a bit of space in between. Now on my picture, I have exaggerated this space, but absolutely positively there is this space in here. Where can I put this? I can sneak it in right here. This space is what is known as Bowman space. And these three things together, the glomerulus, Bowman's capsule, and Bowman's space, collectively are known as a structure known as the renal corpuscle. So the renal corpuscle is a specialized structure comprised of 3 components: Bowman's capsule, or the parietal layer, let's be more specific, of Bowman's capsule, glomerulus, and Bowman's space. Also called glomerular space, which is, again, fine as well. These 3 things together form a structure known as the renal corpuscle. As mentioned, these renal corpuscles are found in the cortex only. If you took me for 430, when we first learned about tissues, the very first tissue we looked at was a kidney. So we could look at this Bowman's capsule and we could see a simple squamous epithelial tissue. And at that time, one of the things I mentioned is that this structure is found only in one place in the body, and that one place in the body is the kidney. Now we can be more specific. These renal corpuscles are found in one place in the body and one place only, and that is the cortex of the kidney. So these three things work together to form that component, that specialized structure in the cortex of our kidney known as a renal corpuscle. All right. Questions on that? All right. So let's kind of work our way out from here. Obviously, some, but not all of the blood, whoops, that's not going to work, is filtered out. So, of course, as we know, we'll sneak it around this way. Leaving the glomerulus is going to be our efferent arteriole. If I do that, I can sneak it right here. Perfect. Efferent arteriole going out. And the same thing is true for our filtrate. While the filtrate needs to be collected by Bowman's capsule, it is not going to be just stay its entire time in Bowman's capsule. It is going to leave. And as it leaves into this tubule that is very twisty and turny. This twisty and turny tube happens to be very, very close to the renal corpuscle. And so as a result, it is called the proximal convoluted tubule. Proximal because it is closest to the renal corpuscle. Convoluted because it is all twisty and turny. And tubule because it is a tube. Of course, being a tube, it needs to be made up of some type of epithelial tissue. And this tubule is lined by a simple cuboidal epithelial tissue. We can draw one of those right here. Right, simple cuboidal cell. Now, this proximal convoluted tubule, actually, let's do this, is where most of the processing takes place. So, this simple cuboidal epithelial tissue has a couple specializations to them. The first is we would want to increase the surface area of the simple cuboidal so that lots of reabsorption and lots of secretion could take place. What is a way that we could increase the surface area of this cell to help in this functioning?
Microvilli?
Exactly. Massive, extensive microvilli. And those microvilli are going to increase the surface area. Now, when it comes to sorting what material gets back into the blood and what stays in the urine, do we want to do that randomly or do we want to be very selective about what comes back and what doesn't? Yeah, we want to be selective. And if we're going to be selective, what is typically going to be required for having that type of specificity, that type of selectiveness? Are we going to allow things to be able to just passively diffuse back and forth? No. What are we going to need? If we're going to direct something specifically, controlling it, so it's not going to be passive, it's going to be active. What do we need for active transport? Yeah, ATP. So we're also going to have lots of mitochondria. So that, and again, I'll, as best as I can in this tiny little space, draw a couple of those kidney bean shaped mitochondria in there for the massive amount of movement that we're going to want to take place. So if we're going to actively do it, we need lots of ATP. So we are going to, and I'm going to need some of this room back. So sneak that up and sneak that up. Excellent. So we have this big twisty turny tubule that then changes orientation down to form this long hairpin turn where it goes, straightens out for a bit, takes a major hairpin turn, and comes back again. This is what is commonly referred to, or is referred to as the nephron loop, but of course it has a fancy anatomical name to it, the good old loop of Henle. Because good old Bob Henle was the first person who identified this. Now, there are some key characteristics about this loop of Henle. Obviously, there is a portion of the loop of Henle that travels down. That's way too big. And a portion of it that comes up. So when we talk about the loop of Henle, we have both a descending limb and of course then an ascending limb. Some, not all, but some of our loops of Henle are very long. So long that they travel all the way out of the cortex and go deep into the medulla. In fact, if you remember, we talked about the medulla having a striped appearance. It is these long parallel limbs of the loop of Henle that actually give the medulla its striped appearance. Now, obviously, both of these ascending limbs and descending limbs carry filtrate in different directions. One brings it down, one brings it up. But they also have different functions as well. And so not surprisingly, they have different anatomies as well. The the descending limb is also known as the thin limb because it is comprised of simple squamous epithelial tissues. So this is the thin descending limb, comprised of simple squamous cells. So not surprisingly, if we've got one thin and descending, then the other must be the thick and ascending limb. And so the ascending limb is the thick limb, and it is made of simple cuboidal. Similar to the proximal convoluted tubules, but fewer mitochondria and fewer microvilli. Let's say it this way, fewer microvilli. Maybe not fewer mitochondria, but fewer microvilli. And we'll draw some cube cells over here. All righty. I am guessing you can figure out what happens next by the names. Anytime we have a proximal something, what else are we going to have?
Distal.
A distal something. And so lo and behold, we are going to have a distal convoluted tubule. So once again, twisty and turny. This distal convoluted tubule is where our fine tuning takes place. Typically, this is where we would tell the kidneys, hey, we need to hold on to more water, or we need to hold on to more sodium, or we need to get rid of more calcium. And how do we normally tell the kidney we need more calcium, or we need less calcium, we need more sodium, or we need less sodium, we need more chloride, or we need less chloride? Hormones, exactly. So typically this is going to be driven by hormones, hormone-driven fine tuning. Now, obviously, we're fine tuning. We're not going to require quite as much movement, quite as much energy. So not surprisingly, this is indeed a simple cuboidal epithelial tissue, but this has far, far fewer microvilli and fewer mitochondria. So we can slap one of those in here as well. Little cuboidal shell. So much fewer mitochondria, much fewer microvilli. From here, our filtrate exits the nephron and enters into a separate structure. So we'll draw this structure in a lighter green to again emphasize that it is not a part of the nephron. And the filtrate enters into a structure. Let's do this. Move this up here. Exits the nephron. Enters into the collecting ducts. The collecting duct travels through the pyramid to the papillae. And expresses the urine at the end of that. However, it is still filtrate in the collecting ducts because it can also fine tune. So there is some processing that is going to take place. Let's say it that way. So the filtrate can still be processed in the collecting duct. Even though the collecting duct is not a part of the nephron, it is capable of some processing And so it is still considered filtrate until it finally gets to the end and is released out the papillae. And once it is released out the papillae, then it is finally considered urine. So 4 important tubular structures. Oh, and I guess the one thing I should say, the collecting duct is made primarily of 2 types of cuboidal cells. To make this small so it fits, I have room to write for my blood vessels. So they're both cuboidal cells. And they are principal cells and intercalated. And when we get to the collecting duct, we'll talk about those 2 cells and talk about what they do. All right, excellent. As you can see, I have done a truly amazing job of drawing this. Again, I am just a regular Leonardo DiCaprio. And here we see the pretty picture from your textbook that shows this as well. So notice it starts with our Bowman's capsule wrapping around the glomerulus. And again, notice that parietal layer is those simple squamous cells just forming the cup that is going to collect the fluid. However, remember there are these special foot-like processes, these podocytes that sit on top of the glomerulus forming the visceral layer. Filtrate exits Bowman's capsule into our proximal convoluted tubule, where most of the processing takes place. So large number of microvilli, large number of mitochondria. Goes into the loop of Henle, where we have our thin descending segment made up of simple squamous cells, and our thick ascending end made up of cuboidal cells. And notice the cuboidal cells of the thick ascending limb are similar to those in the distal convoluted tubule. Fewer microvilli, fewer mitochondria. They then exit the nephron into our collecting duct, which has principal cells and intercalated cells. And we'll talk about the different functions of those 2 when we get to it. So we've talked a little bit about function, but we've identified all the structures and identified the tissue types they're comprised of, which is going to be important because we're going to need to be able to distinguish these under the microscope. And we absolutely positively will do that. We'll do a little of that today. All right. Questions on this?
On the collecting duct,
Yes.
there's all like those other little tubes coming off of it. Is that more convoluted tubules that will connect there, or what connects?
Exactly, yes. Collecting ducts, as their name indicates, is going to collect filtrate from many nephrons. Remember, we've got over 1 million nephrons in a single kidney. We're not going to want each one to have its own collecting duct. So a collecting duct can collect filtrate from hundreds, if not thousands, probably more likely thousands of nephrons, all feeding into those same collecting ducts. All right. And again, the 2 points I want to emphasize is the collecting duct is not a part of the nephron, but remember, it still can actually play an active role in filtering the fluid. So it's not urine until it leaves the collecting duct. Once it leaves the collecting duct out of the papillae into the calyx, then and only then are we done processing it. And so then and only then is it urine. All right. Excellent. But we're only halfway through with our nephron. So let's go back to my glorious picture. where we have here these different, and I'll cheat and grab all these and bring them out of our way so I can bring them up as I need them. Let's talk first about the glomerulus. The glomerulus, as I mentioned, is a specialized capillary. And there are a couple things that make it very special. One is that it is a high pressure capillary. Right, normally we think of capillaries as being relatively low pressure, and I will actually give you the values of this, and we'll talk about the values of this as well. One of the reasons it is high pressure is because, as we mentioned, it is both, let's say it that way, both fed into and out of by an arteriole. Normally, arterioles feed into capillaries and venules feed out. But there is a third way that our glomerulus is specialized. The third way, normally when we think of a capillary, we think of exchange. As we saw in a typical capillary, at the arterial end, things come out. At the venous end, things come in. So capillaries are where materials are exchanged. But that's not the case for the glomerulus. In the glomerulus, this is only involved in filtration. And because we talked about this in cardiovascular, filtration is that out of the capillary or into the capillary? Is filtration when things move out of the capillary, or is filtration when things move into the capillary? I thought this would be an easy question. Well, what's the opposite of filtration? So glad this is a cumulative class that you guys have a final exam in May 19th. It is April 14th, a month and 5 days. Awesome. Well, what if I told you that the opposite of filtration was reabsorption? Then would filtration be out of the capillary or into the capillary? Out.
Out.
Excellent. Only involved in filtration, only involved in coming out, which if you notice with the arrows over here, I kind of already told you earlier, right? This is where we're making the 200 liters of filtrate. They're coming out of the glomerulus into Bowman's capsule. All right. Now, as I mentioned, the glomerulus feeds into an arteriole, and that arteriole is the efferent arteriole. And being an arteriole, basically, as we talked about, like all arteries, it's a conduit, it's a passageway. It carries blood from the glomerulus to the peritubular capillaries. Bing. End of story. Now, with a name like peritubular capillaries, guess where they're located? Yeah, peri means around, tubular means tubes. So these basically wrap around the tubules of the nephron. Oops. So oops. They wrap around the tubules of the nephron. And of course, as we know, they are a typical capillary. So, unlike the glomerulus, they have normal low pressure. Unlike the glomerulus, they receive from an arteriole. and feed into a venule. So again, feeding out of this is going to be a venule. Oops. Oh well. And that venule, of course, has the clean blood in it. And that clean blood then, of course, enters into our cortical radiant vein. And the other way that the peritubular capillary is a typical capillary is in that there's the two-way movement of materials. So both filtration and reabsorption occur. All right. Questions on that? Oh, and since I put arrows on the last one, let's put arrows here as well. Oh, wow, way too tiny. Green. All right, so again, materials coming in and out of these capillaries. Now, lastly, remember I mentioned some nephrons have very long loops of Henle. And those that have very long loops of Henle also have a special capillary on that long loop of Henle. It is like a ladder-like or kind of draping type of capillary. Let's draw it here in, hold on, I want this dark red. So it kind of drapes down. Let's make it a little bigger. Over this long loop of Henle with this kind of lattice or ladder-like appearance to it. And this long specialized draping capillary is known as the vasa recta. This vasa recta plays an important job in helping to regulate the concentration of the urine, right? We can produce a large volume of dilute urine or a small volume of concentrated urine. Now, is it because we care whether urine is concentrated or dilute? Why would we want to concentrate the urine? Where does that water go if we concentrate the urine? Guys are overthinking this.
The water can get reabsorbed.
Exactly. So really, it's not so much about whether the urine is a concentrated or not. It's really about how much water we put back into the blood. If you are very well hydrated, because you've been drinking your 18 glasses of water a day or whatever it is that you're supposed to drink, right? You're well hydrated. You've got plenty of excess water in your body, fluid in your body. And so you can produce a large volume of relatively dilute urine. However, when you first wake up in the morning, and because again, COVID and 2020 and everything that goes along with that, if you're like me, you've been sleeping for about 12 hours, which means you haven't been drinking for 12 hours, right? I just mean from the depression. I don't mean, you know, I have kids, so I don't sleep for 12 hours, but let's just assume you were sleeping for 12 hours. You obviously haven't drank in 12 hours. So what is that morning urine like? Is it super clear and dilute when you first produce that morning urine? No, it tends to be a smaller volume of more concentrated urine because you've been needing to hold on to more water because you've been releasing moisture in your respiration, you've been using water other ways, and so we need to maintain and hold on to that water. So yes, it plays an important role in concentrating the urine, going the difference between dilute and concentrated urine. But again, remember, it's not the urine we care about, it's the amount of water in our blood. All right. So there you go. There is the pretty picture of our blood vessels. And again, I have done a truly amazing job of drawing this, but the artists from your textbook but do a decent job as well. So let's look at theirs. Again, basically now we know there's really 3 capillary beds. Our glomerulus. Here's that pretty picture. And we'll talk about more and look at more histology in a minute. Our friend and your friend of mine, the renal corpuscle. Here we see that simple squamous epithelial tissue of the parietal peritoneum. Parietal Bowman's capsule. And then this big old huge squiggly thing here in the center is our glomerulus. Notice, and I'm going to go ahead and clear this picture for now, all these nuclei that you see are typically the nuclei of those podocytes that are sitting on top of it. Remember, that's the visceral layer. However, we can see some places where there's some blood vessels in there. I mean, some blood in the blood vessels in there. Here we see it inside of Bowman's capsule, and we can see how it feeds into that paratubular capillary that wraps around the tubules of our nephron. And notice here, we have one of those nephrons with a big, huge, long loop of Henle with that nice, elaborate, draping capillary over the top of it, the vasa recta. Now, the one point I want to make about this particular illustration is the point of this illustration is to show you the difference between a paratubular capillary and a vasa recta. I want to make a point of emphasizing it's not an either or. You don't get one or the other, right? This one with the short loop of Henle can have some of its paratubular capillary wrap around the nephron, and it would still be paratubular capillary. This one over here, just because it has a vasa recta, doesn't mean it won't have a paratubular capillary. These are not exclusive. It's not either or. It's not either or. They're just showing you them separately here because they really want to emphasize the 2 different types. In fact, I think I have a better picture. Well, we'll talk about that one in a second. How far do we have to go?
So do you always have paratubular capillary, but you don't always have vasa recta?
Correct. And notice here's a great example. Here we see a typical nephron with a paratubular capillary that wraps around the entire nephron, including the loops of Henle. Here we see one of those with the vasa recta, but notice it still has a paratubular capillary as well. So that's a great point. Let's go back to our drawing and emphasize this. All, and let's do this big and bold. All, oh, that's not big. All nephrons have a paratubular capillary, but only some, only those few, and actually let's do that. Only those few that have the really long loop of Henle have a vasa recta. So that's a great point. Thank you for making that. All right. So now we have to cheat and go back a little bit. All right, questions on that? Notice, essentially, there are 8 structures that make up a nephron. 4 tubular structures, 4 blood vessel structures. Although really, like I said, 3.5, because only a few have that vasa recta. All right, excellent. I love this picture from your textbook, which of course is always a dead giveaway that it's something that's likely in your test bank. But notice what they've done here. They've done 2 things. They have taken essentially what is a epoxy and injected that epoxy into the blood vessels of the nephron. And then they basically dissolved all the tissue away that surrounds it. So what we're left with is this basically model that we can then look at a high magnification view that shows the blood vessels here associated with them. And in particular, here we're in the cortex. You can see that there are primarily 2 different types of capillaries. Notice one of these capillaries are small and compact. They look a little bit like brains, which may be one of the reasons why I like it so much. We have these tightly packed brain-looking capillaries. And these capillaries are the glomeruli. They're the ones that are compacted because Bowman's capsule wraps around them. Notice the second type of capillary are these big, huge, loose ones with a lot of spaces in between. These are clearly our paratubular capillaries because you can see how there's all that space in between because they're wrapping around the tubules. Now, again, we've dissolved the tubules away, so you don't see the tubules here, but we just see excellent question. You can just see the blood vessel structures. And notice here, let's draw one more thing, because they're the ones that they've labeled. Notice here we have a glomerulus. And so when it comes to identifying the difference between an afferent and an efferent, well, again, it's in the name. Afferent and efferent are associated with the glomerulus. So notice the one that feeds into the glomerulus is the afferent arteriole. The one that feeds between the glomerulus and the paratubular capillary is going to be the efferent arteriole. Of course, that does leave one more question. What's this blood vessel right here that I've just labeled in red?
Is that the cortical radiate artery?
It absolutely is. So the cortical radiant artery feeds into the afferent arteriole, feeds into the glomerulus, feeds into the efferent arteriole, feeds into the paratubular capillary. And notice we can see all of that very, very beautifully with this great microscopic view of this. Like I said, basically it's a high resolution view of a little plastic model that was made by injecting the blood vessels and peeling everything else away, really dissolving all the tissue away. So all you're left with is this beautiful, beautiful model. And like I said, it makes me happy, probably because those glomeruli look like brains, and I like brains. All righty. Questions on that? All right, excellent. Here we see a summary of all these together as we were just talking about. But as we also hinted at, there is one big major difference in our nephrons. That is an excellent question. I don't know if I did. Let's go ahead and do that just to make sure. It's always best to have 2 of it rather than forgetting to do it. So I have definitely saved it now. So excellent. Thank you for asking. 2 main types of nephrons. And as we said, the big difference between these nephrons involves their loops of Henle. Some of them have big, long loops of Henle that penetrate deep down almost all the way through our medulla, whereas some have much shorter nephrons that are either entirely in the cortex or just barely enter into the nephron. Well, not surprisingly, the types of nephrons that are almost entirely contained or are entirely contained in the cortex are known as the cortical nephrons. And those are the majority of them, making up about 85% of our nephrons. However, those closer to the border between the cortex and the medulla, and that's what they're trying to represent for us here, that border between the cortex and the medulla, who have their nephrons that penetrate deep down with their loops of Henle into the medulla, are what are known as juxtamedullary nephrons. And again, much, much fewer of them. These are the ones that will have the vasa recta. And I know 15% doesn't sound like a lot, but remember, in a kidney, we have over 1 million nephrons. So if you have over 1 million nephrons and 15% of them are juxtamedullary, how many juxtamedullary nephrons are there in a single kidney?
150,000?
I don't know, that's too much math, but it's a lot, whatever it is, right? No, you're right, absolutely, that's exactly how much it is. But the point is, it's absolutely a lot. So there's absolutely a ton. So there are a ton of them. Yes, the vasa rectas are only found on the juxtamedullary. And they're the only ones that have that. And like I said, your book has these nicer pictures. Again, here is a cortical nephron. Notice with that cortical nephron, it can have capillaries on the loop of Henle, but it's not that long draping lattice-like nephron. So this is just a paratubular capillary that is over all of it. However, when we get to a juxtamedullary nephron, notice it has a paratubular capillary over the proximal and distal convoluted tubules, but it also has this long draping lattice or ladder-like one over its loop of Henle that is that vasa recta. So vasa rectas are only found on the juxtamedullary nephrons. All righty. Questions on that? All right, we have a bit more anatomy we need to talk about. So again, a lot of vocabulary coming up. So I think this is another good stopping point. Let's go ahead and take our second break here. Looks like it is 10:26. So that means coming back at 10:41. And I will start the recording at that point. So any questions on the anatomy we've covered so far? If the anatomy we've covered so far isn't making sense, it's just going to get worse as we move forward. So now is the time to ask. So any questions on this before we move forward? All right, excellent. Let's go ahead and take our second break here, come back at 10:41, and at 10:41, we will pick up the lecture from here. I will see you guys in 15 minutes. So we've been working our way through the anatomy of the kidney, and we're down to the level of the nephron, and there's a little bit more that we need to go over. Here we have some really nice illustrations that do a great job of showing us some of the anatomy here that we are looking at. Notice here again is our Bowman's capsule, and we can clearly distinguish the difference between our parietal layer, which is the simple squamous, and the visceral layer that has these podocytes. Notice here we see some examples of podocytes. Oh, and that brings me to one more point, one more specialization of our glomerulus. Notice our glomerulus is a fenestrated capillary. Has those holes in it, those fenestrations, which again allow it to be more diffuse in what is able to come out. Bigger holes mean more things can come out of it. However, we don't want too much stuff getting out. So notice these podocytes with their long, extensive foot-like processes that stick out interlock to form those filtration slits that are going to help to regulate what comes out and crosses this membrane. So we see that really nicely right here. And again, the illustration of this makes these cool looking foot cells look really neat, but they can't really look like that, can they? Well, turns out they indeed do. This is a colorized, so the color is artificial, but this is actually an electron microscopy view of the glomerulus where we can clearly see the body of the podocyte and its long foot-like processes with those little foot-like processes that stick out, forming the filtration slits that are going to help to regulate the movement of the filtrate and selectivity of the filtrate as it is coming out of that glomerulus. Here we see an illustration of what this filtration membrane is. Again, this is basically between the glomerulus and Bowman's space. This probably doesn't have to be that big. So as you can see here from the illustration, we have our plasma here inside of the glomerulus, and then Bowman's space would be out here. And notice basically this filtration membrane is made up of two components. The simple squamous fenestrated endothelium of our capillary, the foot-like processes of our podocyte, and basically a thin layer of basement membrane that holds these two structures together. So we have this thin filtration slits that allow for movements. Notice we're going to be able to get obviously water, ions, all sorts of small molecules and other things that are be able to pass through here, but large proteins and most importantly our formed elements, our erythrocytes, our leukocytes, and our thrombocytes cannot pass through. So they are going to stay in the capillary. All right, so again, we're going to have that filtration that occurs there. Now, there's one more point, and I'll go back quickly to our illustration that I drew, because I think it's still here. Absolutely. Excellent. If you were paying close attention, and again, I drew an awful lot on this, so it's totally okay if you didn't. But if you were kind of paying close attention, one of the things you may have noticed is that I had a corner of our distal convoluted tubule come right up here next to our renal corpuscle. And this was not on accident. Because if you remember, when we were talking about the lungs, we talked about in between every individual alveoli and every individual pulmonary capillary, we have some intrinsic controls. Because while we say the oxygen carrying, probably not the oxygen, the partial pressure of oxygen is 104 in the lungs, it's not truly 104 in every single individual alveolus. There's going to be changes. And the same thing's true here. We need to be able to make dynamic changes in every single one of those 1 million nephrons to make sure that they are being efficient. And we do that with a very specialized regulatory structure associated with the renal corpuscle, and I want to emphasize, the distal convoluted tubule. So between the two of these things, we get this specialized regulatory structure that is known as the, oops, where's my, oh, there it is. Okay, we'll get to it in a second. Not sure what all of that's supposed to be. Collectively is known as the juxtaglomerular apparatus. Here, we clearly see Bowman's capsule, glomerulus, podocytes, so Bowman's space. So again, we have that renal corpuscle. Notice we have our afferent arteriole feeding into the glomerulus and our efferent arteriole feeding out. And the other thing we see here is a bit of our distal convoluted tubule. However, notice there's something interesting going on here in our distal convoluted tubule. This interesting thing that's going on here is that we said it was simple cuboidal in nature, but if you look closely, right here, where it is in close proximity to our afferent arteriole, is this tightly packed cells that become simple columnar. And these simple columnar cells are known as macula densa cells. Macula is focal, densa, dense, so it's densely packed cells in this small location. These cells basically are our quality control. Notice they're in the distal convoluted tubule. Which, as we mentioned, is only involved in the fine tuning of the filtrate. Most of the work has been done. So in the distal convoluted tubule, what we have is essentially mostly urine or almost urine. Still going to be a little bit of fine tuning, but at this point it's almost urine. And this almost urine is a perfect time to check it to see, are we processing it at appropriate amount? In my mind's eye, I always picture that old I Love Lucy. Many of you may have seen it, where she's sitting working at the candy factory. And her job is the candies come down the conveyor belt and she's supposed to take the candy and put it inside of the little paper. And at first she's able to do that very efficiently because things come, yeah, classic episode, absolutely, come relatively slowly, but then they start coming faster and faster and faster and faster, and she can't keep up. She starts throwing them down her shirt and throwing them in her mouth and doing all sorts of other things with them. And that's what's happening here as well, right? And it doesn't just happen at I Love Lucy. Maybe you haven't seen any black and white TV because you're all children. But maybe back in ancient times when we were allowed to leave the house, maybe you went to the Jelly Belly factory down in Vacaville. Right. And one of the things that they have on that machine with the Jelly Belly factory is when you're there at the Jelly Belly factory, they have all of the bad jelly beans that have been filtered out in the process. Anyone know what they call those?
Flops.
Belly
Yeah, they're the
flops.
belly flops. Absolutely. Well, that's basically what our macula densa is. It's the quality control. It's looking at the filtrate that is there for two things. Notice if we are making filtrate too fast, like that conveyor belt is coming too fast, then it's going to be under processed. We're not going to be able to keep up. However, the opposite is true as well. If that Jelly Belly factory, every single Jelly Belly came down one at a time, you could really micro inspect them, and really, even tiny minor flaws might cause you to chuck it out. So notice if we're making filtrate too slowly, it's going to be over processed. So the job of the macula densa, and I don't think I wrote this here, it contains chemoreceptors and osmoreceptors. So we can check the chemical composition and also the osmolarity, just the amount of stuff that is in it, and check to make sure we're producing filtrate at an appropriate rate. Not enough is bad, but too much is bad as well. It needs to be at an appropriate rate. So this is where quality control takes place. All right, we comfortable with that idea?
Is that distal tubule that belongs to that Bowman's capsule, or is it possible it's from a different?
No, it's
one?
so again, if we cheat and go back to my drawing, notice it is this part of the distal convoluted tubule. So here in the distal convoluted tubule are going to be those tightly packed cells that are going to be close to this, and also close to the afferent arteriole. And we'll see why it needs to be close to those two things in just a second. So notice if we were tracing the path of our urine or the filtrate, it comes out of the glomerulus into the Bowman's capsule, into the proximal convoluted tubule where most of the processing takes place, into the loop of Henle, right? Back out the distal convoluted tubule, and near the end of the distal convoluted tubule, where it's almost done processing, we now can do our quality control. All right, so it is a part of the distal convoluted tubule, and that's what they're trying to represent in this picture. Of course, now that I go back, it's all going to be gone, but that's all right. This is a little bit of that distal convoluted tubule close to the renal corpuscle, not a part of the renal corpuscle, just close to it. And again, this contains our chemoreceptors and osmoreceptors to check our rate of filtration. Now, obviously, checking the quality is important, but if it's off, you need to let people know. And so it releases chemical signals. And one of the primary chemical signals it releases is nitric oxide. Not nitrous oxide, that's what you get at the dentist's office or at that rave on Friday night, right? Nitric oxide, but nitric oxide works in a similar fashion. Nitric oxide, one of the things that it does is it is a vasodilator. So if you release more of it, what happens to a blood vessel? Dilates it, exactly. It dilates the blood vessel, and if it dilates the blood vessel, more blood comes to the area. And if more blood flows, basically filtration rate is going to increase. On the other hand, if you release less nitric oxide, our blood vessel constricts. And if it constricts, less blood flows. And if there's less blood coming to the nephron, filtration rate decreases. So it turns out these macula densa cells speak primarily to the afferent arteriole. And not just any part of the afferent arteriole, but these specialized smooth muscle cells on the surface of the afferent arteriole. These cells are called granular cells. because they're smooth muscle cells with vesicles that release hormones. These granular cells are also known as juxtaglomerular cells. So notice, and here I'll erase all this now. Macula cells, chemo and osmo receptors, they release that nitric oxide to communicate with our granular cells, or what are also known as juxtaglomerular cells. If you use juxtaglomerular in an essay question, for instance, once you've spelled it out once, you may call them JG cells. But these are the special cells, special smooth muscle cells on the afferent arteriole. And I say they're special smooth muscle because they are both smooth muscle cells and endocrine cells. They're smooth muscle cells, so they can contract and dilate to change the diameter to affect the flow of blood into the glomerulus, but they can also release hormones. This is where the erythropoietin is released. This is where renin, our hormone-like protein, is released, right? So this is where that work is done. So that's why these are granular cells. They have vesicles containing these hormones that can be released into the blood to communicate with the body. But it turns out these juxtaglomerular cells are not the only cells that the macula densa communicates with. The other cells that the macula densa communicates with are these cells that you can see are kind of interdigitated in between the glomerulus. These cells are called mesangial cells. These mesangial cells are very dynamic cells. And they contain a large amount of actin. Now, normally we think of actin in terms of contraction. We learned in our skeletal muscle, for instance, how it contracts. But if you remember, actin is a cytoskeletal structure that allows a cell, yes, to contract, but it also allows the cell to dynamically change its shape. What this cell is able to do is it can rapidly build up or break down actin filaments. Now, if it builds up actin filaments, adds to its cytoskeletal, adds to its structure, and as it does that, it causes the cell to expand. So it's adding more skeleton to it, basically, and it causes the cell to expand. Now notice, this is surrounded by, let's say it that way, the glomerulus. The glomerulus is this big clustered capillary. If the cells underneath it start to expand, it pushes out the capillary of the glomerulus. So as it expands, it pushes the capillary out, or let's be specific, pushes the glomerulus out. And as you expand the surface area expand this glomerulus, it expands the surface area. And when you expand the surface area, more diffusion takes place, so we can make more filtrate. Conversely, if we break down actin, it causes this cell to collapse. And as the cell collapses, the glomerulus shrinks. And as the glomerulus shrinks, less surface area. And with less surface area, less filtrate is made. So these three cell types work together to help to regulate the making of filtrate, the processing of the blood. The picture is important. We need to know it. We need to recognize, we need to understand it. But I think it's also important to understand this process. So let's take the picture out of it. Let's talk about the function of the juxtaglomerular apparatus. Right? Again, it is comprised of three types of cells. Oops, I liked that big. Look at that big. Those three types of cells are, well, actually, let's do it this way. The macula densa cells. Again, these cells are located in the walls of the distal convoluted tubule. Right? And they contain chemoreceptors and osmoreceptors. Cell 2 are the juxtaglomerular cells. Or what we can also call granular cells. And again, granular cells is an appropriate term for this in this case. I will accept granular cells. Of course, if I ask you the function of the juxtaglomerular cells, you need to know what that term means. But if you're writing this out on an essay question, saying juxtaglomerular or granular, either would be fine. These are located in the walls of the afferent arteriole. And their function is they are, let's say, combo of smooth muscle and endocrine cells. So their job is to change the diameter of the afferent arteriole and release hormones. And the third cell are the mesangial cells. These are located not in the glomerulus, but under the glomerulus. A little more of them here. They dynamically change their size using actin. And by doing so, they can change the surface area of the glomerulus. So those are the three types of cells that make up the juxtaglomerular apparatus. And let's walk through the process. Let's say, for instance, that our macula densa cells detect that we are over processing the filtrate. So if we're over processing the filtrate, what's the problem? Are we making too much filtrate or are we making too little filtrate?
Too little?
Yeah. So our macula densa is going to send a signal to the afferent arteriole, or more specifically, to the granular cells. And what do we want the granular cells to do?
Send signals to constrict.
So if we constrict, is more or less blood going to go to the glomerulus?
You want to dilate, some more goes.
Yeah, we want to dilate in this case. We want more blood going to it. So we want the afferent arteriole to dilate. So how would we do that? Is our macula densa cells going to send more nitric oxide or less nitric oxide?
More?
More.
Yeah, more nitric oxide to the afferent arteriole, and that nitric oxide causes the afferent arteriole to dilate, more blood to the glomerulus, and we make more filtrate. But we also, our macula densa cells, need to send a signal to the mesangial cells. What do we want the mesangial cells to do?
Those are the ones that change their size, right?
So do we want these cells to get bigger or do we want these cells to get smaller?
Bigger?
Yeah. So we want the cells to make more actin, and that makes the cells bigger. Because you're right. If the cells get bigger, they push up on the glomerulus. Right? Think of it in terms of my hand. I've got my hand clustered together into a fist. It has a relatively small surface area. But when I spread my fingers out, I now have more surface area. So by pushing the glomerulus out, by spreading the glomerulus up, we get more surface area. We get more surface area to the glomerulus, and if we get more surface area to the glomerulus, it can make more filtrate. And by both of those acts, we have solved our problem. Our problem was we were making too little filtrate, so we bring more blood, which is going to cause us to make more filtrate, and we spread out the glomerulus, which is going to cause us to make more filtrate. And now we're going to be producing more filtrate at a more appropriate rate. However, part of the problem is maybe I opened up the floodgates too much, and now suddenly the macula densa cells detect that we are under processing the filtrate. What problem does that indicate?
Too much filtrate.
Yeah. So let's work it out. Our macula densa cells are sending a signal to the afferent arteriole. And again, specifically the granular cells. And in this case, what is the signal going to be? Are we going to send more nitric oxide or less nitric oxide this time?
Less.
Yeah. Exactly. We're going to send less nitric oxide. And when we send less nitric oxide, as we know, that is then going to, so less nitric oxide causes the afferent arteriole to constrict. So when it constricts, less blood to the glomerulus. And therefore, with less blood, we make less filtrate. We've been able to bring our filtrate rate back down from being too high. Of course, as we also mentioned, our macula densa cells are also send a signal to the mesangial cells. And what do we want the mesangial cells to do this time?
Reduce the surface area.
Yeah, we want to break down actin. When we break, I agree entirely. Exactly. When we break down the actin, the cells get smaller. Right? I could not have said it better myself. Absolutely. The cells get smaller as a result of that. And when the cells get smaller, the glomerulus collapses down. And as it does that, it has less surface area. And with less surface area to the glomerulus, it can make, oops, I'm trying to cheat and use the same wording that I used in the last one, less filtrate. There you go. So if we take the picture out of it for a minute and just think of how these three cells work together, we can see that it is a very simple but very elegant process. And that's the beauty of this. Every single nephron has its own, right, distal convoluted tubule, its own macula densa. So every single nephron is able to communicate with its very own afferent arteriole to make sure that every single nephron is making filtrate at the appropriate rate. We don't just have one global control for all of them. There are global controls that influence all of them, but as we know, everyone's going to work at a slightly different rate. And so being able to make those minute adjustments in these specifically allow for that intrinsic control, right? That's this magic word we've used many times so far. Right? This intrinsic control from within the organ itself. Each nephron is able to regulate its own rate. All right. Now, I want to show you two more things. Again, notice here we see the illustration that shows this. But if we go to, where do I want to go? I want to go here. Again, as we go to our modules, one of the places you can go with our modules is to that CRC Virtual Anatomy Lab. When we do that, of course, there is a urinary system. And I believe I mentioned this in the last class, where there's these great pictures and models, but notice there's also this trifold plaque. Notice here, we can see all of the things that we were talking about in the gross anatomy. Notice we can see, and luckily, again, it's got some nice labels. So notice it's shown us a lobule, it's shown us a renal column, the papillae, the pyramids, the cortex, right? The minor and major calyces, all those things we've talked about. But notice it also does a really good job of showing all the blood vessels we've been talking about as well. Notice the second component of the plaque shows a piece of one of the lobes. Notice we can clearly see here where the arcuate arteries and veins are located. So we have the difference between our medulla down here and our cortex up here. And notice we have some other nice fine details. Cortical radiant artery, afferent arteriole. Notice here we've got our renal corpuscle in its entirety, but this one happens to be open, where we can see the glomerulus. We can see the efferent arteriole feeding out, where it would feed into the paratubular capillaries, into a venule, into the cortical radiant vein, into the arcuate vein. So notice we see all the blood vessels. Notice, again, they've separated the artery, the tubular structure from the blood vessel structure, so it's easier to see it. Here up here, we see the paratubular capillary, and here down here, we see a vasa recta. Notice we have a cortical nephron and all of its components, Bowman's capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, which notice conveniently enough comes here to help form our juxtamedullary, juxtaglomerular apparatus into the collecting duct that goes down to our papillae. And over here, we have a nice juxtamedullary nephron with its super long loop of Henle that goes all the way down deep into our medulla. And another vasa recta over here as well. And lastly, so again, all those great pictures there. Notice here, we see a renal corpuscle, and we see most of our juxtaglomerular apparatus. So notice first, we have, again, our parietal layer of Bowman's capsule, simple squamous cells that notice do a nice job of feeding into our proximal convoluted tubule, which they've even put some microvilli on for us, which is super cool. Notice they show us the fenestrated capillary that is the glomerulus, but they've also done a nice job of putting the podocytes on top of it. So we can see those podocytes that are forming the visceral layer of Bowman's capsule. Now, again, podocytes cover the entire glomerulus, but they've removed part of it so we can see the capillary inside. Notice here, we can tell the difference between the afferent and efferent arteriole, because notice our afferent arteriole has these enlarged, exaggerated cells that represent our granular cells. Here, we can see the simple columnar cells that form the macula densa. Notice what we're missing is in this space in here should be where all the mesangial cells are. So notice our model doesn't show us the mesangial cells, but we can at least see the spaces where they would be located. And this does a really nice job of showing that. However, notice also, if you go back to your modules, Nope, I guess I have to clear the picture. Under this section, we see some histology. And one of the histology that we've got here shows us that distinction between the medulla and the cortex. Remember, we have lots of good websites. That Yale site is really excellent for that. Here, we actually see a close-up view. And again, I didn't do the labels of this, but I think this is a great picture that does an excellent job. If I click on this, do we get to see this big? Nope. What if I open it? That doesn't help me. That's not any bigger. Are you guys seeing that? Is
The
it
screen
still-
didn't change.
It didn't enlarge for you guys?
No.
Okay, well, you can do this on your own. Can you see my pointer on it? Okay,
Yes.
so there's a couple things here. Actually, I'm going to use color because it stands out more. Notice we can clearly see the space that is Bowman space and that renal corpuscle. The renal corpuscle tells you you're in the cortex of the kidney, and it is a super obvious thing. We can see our glomerulus, we can see Bowman's capsule, we can see Bowman's space, we can see all of that very, very clearly. But there's two other things that I want to show you on this as well. The first is for the renal tubules. Remember, we talked about how both the distal and the proximal convoluted tubules are simple cuboidal. But one of the big differences in them is the presence of microvilli. So notice when we look inside some of these tubules, they look very dirty on the inside of them. The reason they have this dirty appearance to them is because they have large prominent microvilli on them. So those are the proximal convoluted tubules. However, notice some of them are very clear on the inside. And the ones that are very clear on the inside have far fewer mitochondria. They don't have none. There's still a little bit of fuzz in there, but not none, but much less. And those are typically how you're going to distinguish the distal convoluted tubules proximally. However, notice something special about this particular distal convoluted tubule. While many of its cells are simple cuboidal, here we have this tightly packed together, almost columnar cells right next to the glomerulus. We can actually see the macula densa histologically. And you absolutely positively are going to be responsible for that macula densa histologically. Now, if I close this, whoops, I don't know what I just did. I think I have one more good picture as well. Excellent. This picture, and again, I know you're not going to be able to see this as high magnification on my screen, but I'll still enlarge it for me. The cortex and the medulla are pretty easy to distinguish. Notice nowhere. I did download it. This is the downloaded version that I'm trying to use here. But again, I think it's limited by what I'm able to show from my page. I wonder if I share my screen, do you guys see it big? Okay, excellent.
Now it's there.
Perfect. So notice two key things about the medulla. Notice nowhere in there is a renal corpuscle. So the fact that we don't see a renal corpuscle is a dead giveaway that we are in the medulla. Notice also in the cortex, we have the proximal and distal convoluted tubules. So all the tubules are going every which way. However, remember in the medulla, the collecting ducts, the loops of Henle, all of those things pretty much run parallel. So notice here, the tubules are much more uniform in their orientation. Now, I'm not going to hold you responsible for being able to figure out which of these tubules is a collecting duct, which one is the thin descending, and which one is the thick ascending. But one of the things you will notice in the medulla is in the medulla, you will see these capillaries, these blood vessels that run parallel to all the tubules. And guess what these capillaries that run parallel to all the tubules are? Yeah, if you're not sure, hopefully you could at least read it off the screen now that I've made it big. They're the vasa recta. So in the medulla, not only can you see the fairly parallel tubules, but you can actually see the vasa rectas here as well. So if you look at your histology list, these are the types of things from this anatomy that you are responsible for. And I'm pretty sure everything we mentioned was on the list. All right. Excellent. Questions on that? All righty. Then let's do this. Oops, let's do this. All right, so that is, I think we're sharing, am I sharing the screen again? No, I'm still sharing the screen. Let's, no, that's what I want. Yeah, perfect, sharing this now. All right, so with this, we have done both the gross and the microscopic anatomy that you are going to be responsible for. So that is everything that I wanted to talk about for today. And so that leaves us with anatomy out of the way. So again, we've had our one lecture, again, just exactly like what we did with the respiratory system. One lecture on the anatomy, and then that's going to give us 2 lectures on the physiology. All right, questions on that? All right, I'm going to go ahead